IDL is internalized and degraded to its primary constituents. This is one fate. The second fate is to be converted into a more mature particle, which we call LDL, or low density lipoprotein. There are several events that take place in order for the IDL to be converted to LDL. This depends on the interaction again with high density lipoprotein. So the IDL interacts with the nascent high density lipoprotein. It is composed mainly of phospholipids and ApoA1. A liver enzyme that's called LCAT stands for lecithin cholesterol acyl transferase. So in effect, it transfers an acyl group from lecithin, which is phosphatidylcholine, into cholesterol. So cholesterol is going to be esterified into cholesterol ester, and lecithin is going to be converted into what's called lysolecithin. The cholesterol comes from the IDL to the surface of nascent HDL and react with lecithin in a reaction that is catalyzed by the liver enzyme LCAT. LCAT is activated by ApoA1. So ApoA1 on the nascent HDL is going to activate LCAT, so it is going to transfer an acyl group from lecithin to cholesterol. So cholesterol is going to be converted into cholesterol ester, and lecithin is going to be converted to lysolecithin. Most of the cholesterol ester which is generated is going to be uptaken by the nascent high density lipoprotein, so that the nascent high density lipoprotein, which essentially doesn't have cholesterol or cholesterol ester as part of its constituents, is now going to be converted to other species of HDL. We call them HDL2 and HDL3 and so on. So these other species of HDL now contain cholesterol ester, but part of this cholesterol ester is going to go back to the IDL in its conversion to LDL. So the resulting LDL will now be stuffed with cholesterol ester. In effect, in the conversion of IDL to LDL, its cholesterol content is converted to cholesterol ester to be embedded in the core of the LDL. So now the LDL will have cholesterol ester as the main constituent of the core. Already triacylglycerol has been very much reduced by the action of lipoprotein lipase. But this is not the end of the story for triacylglycerol. There is one more event that is going to reduce triacylglycerol even further. We have another enzyme which is called hepatic triglyceride lipase. This hepatic triglyceride lipase will hydrolyze triacylglycerols even further. So we'll end up in the low density lipoprotein with a very much reduced amount of triacylglycerol, and it will be really stuffed with cholesterol ester. So the cholesterol ester content increases a lot, and triacylglycerol will be reduced very much. On the surface, the LDL particle has a single copy of the ApoB100, and it is going to lose ApoE. We'll end up with a single copy of B100 on the surface of the LDL. The E is going to be lost. Already, C2 has been returned to the high-density lipoprotein after lipoprotein lipase has been stimulated and hydrolyzes triacylglycerol into fatty acids that are stored in the adipocytes in a previous step, as we mentioned before. Then what is the fate of the LDL? LDL is taken up by nucleated cells, including the liver cells. This implies that cells which are not nucleated, like the mature RBCs, do not have LDL receptor and don't uptake LDL. So this is the LDL receptor. It binds the B100 apoprotein. Specifically, it binds the carboxylic domain, which is specific to B100, which, if you remember, we mentioned that it is missing in the B48 of the chylomicrons because of this event of RNA editing, which introduces a stop codon nearly in the middle of the ApoB transcript. This is why ApoB48 doesn't have this carboxylic domain, which is the ligand of the LDL receptor. So the LDL, with its cholesterol ester content, will bind to the LDL receptor on different cells. We consider here the liver cell. And the whole complex of the LDL together with the receptor, they are internalized in a clathrin pit. Then in the endosome, the pH is low. So the receptor will separate from the LDL. Most of the receptor particles are going to be recycled. They go back to the cell membrane in order to bind to another LDL receptor and so on. But the LDL with its constituent of cholesterol ester is going to be degraded. This means that the constituents of the LDL is going to be released, and this will include cholesterol. Only a small part of the receptor is lost. And this is compensated for by transcription of the LDL receptor gene. You have to notice that we started with the VLDL, which was stuffed with triacylglycerol, and in its conversion to IDL and then further to the LDL. Now the main component becomes cholesterol ester. We end up with cholesterol as one of the constituents of the degraded LDL particle inside the liver cell. We have different sources of cholesterol. This is one of them. It comes from the internalized LDL particles and also from the internalized IDL particles. The other external source is the chylomicron remnant source, which comes originally from the diet. So these are the two sources of externally derived cholesterol. But remember, we have de novo synthesis of cholesterol taking place in the liver cell, in the cytosol as well. And there is an interplay between the externally derived cholesterol and internally synthesized cholesterol in order to keep the cholesterol level inside the cell within reference ranges. The regulation of cholesterol level inside the liver cell takes place primarily by regulating the de novo synthesis. We know that the de novo synthesis of cholesterol inside the liver cell takes place in the cytosol in a multi-step reaction that is catalyzed by many enzymes and the rate-limiting enzymes HMG-CoA reductase. So regulation of HMG-CoA reductase is a major determinant of the de novo synthesis of cholesterol, and in turn a major determinant of the level of cholesterol inside the liver cell. So externally derived cholesterol is going to downregulate the synthesis of HMG-CoA reductase if it is high. So if the cholesterol level is high, it's going to downregulate the de novo synthesis of cholesterol through downregulation of the enzymes, especially the rate-limiting enzyme HMG-CoA reductase. And this takes place at two levels. One of them is at the level of gene expression, and the other level is at the level of the enzyme degradation by ubiquitination followed by proteasomal degradation. Also, the cholesterol, if it is high, it's going to also downregulate the transcription from the LDL receptor gene. So eventually, the receptor level will be low. A third mechanism is that if we have high cholesterol level, so this is going to upregulate the enzyme ACAT. So the ACAT of the liver is going to be upregulated, is going to be stimulated. So it's going to transfer an acyl group from acyl-CoA to cholesterol. So it will convert cholesterol in effect to cholesterol ester. The action is similar to the LCAT. So we have ACAT and we have LCAT. ACAT functions within the cell. The ACAT in the enterocyte, if you remember, functions inside the cell. The liver ACAT also again here functions inside the cell. The LCAT catalyzes the reaction on the surface of the high-density lipoprotein, as we mentioned. So these are differences between LCAT and ACAT. LCAT transfers acyl from lecithin. ACAT transfers acyl from acyl-CoA and releases CoA and converts the cholesterol into cholesterol ester. So in effect, this cholesterol is going to be converted to cholesterol ester. Cholesterol ester is stored. It doesn't take part in the cell membrane. So this is one way to control the role of cholesterol in determining the fluidity of the cell. We don't want cholesterol to perform this function in an aggressive way. We want to put it under regulation. We want to put this function of determining the fluidity of the cell membranes under regulation. So if the cholesterol is very high, it's going to dangerously affect the fluidity of the cell membranes. So in order to control that, we convert it to cholesterol ester to be stored and to limit this effect.